Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome to the another class of the course Structural Biology. We are going through the module of introductory section, and today, as told in the previous class at the end, we are going to discuss about decoding biological macromolecule. So, how to decode biological macromolecules? The first and foremost requirement is to understand biological macromolecules is to read them. You have to know about their basics. You have to read how the monomers are forming polymers to start knowing about. We are targeting three polymers: carbohydrate, protein and nucleic acids, and then we are also interested in knowing about lipid and fat. So, how the primary sequence of those polymers could be studied? What are the challenges in studying them? What are the possibilities of improvement where in terms of instrumentation, in terms of technical development, people are working? We will try to make a preliminary discussion. This is not the core topic of our study, but I sincerely believe that if you do not know how to read the basics, you should not go to the higher level. So, let us start with reading carbohydrates. Carbohydrates, as I told, are considered as a very important molecule, because look at the food sources and you will get the most accessible sources. They are very rich in carbohydrate. And if you look at, as we have discussed in the previous class, carbohydrates are connected through glycosidic bonds. A glycosidic bond or glycosidic linkage is a type of covalent bond that joins carbohydrate molecule to another molecule. It might be carbohydrate or it might be others. So, basically what happened in case of carbohydrate, you have a hydroxyl group which is connected to the carbon, and you have another carbohydrate, if we consider carbohydrate, containing the hydroxyl group. Now, it will go through a dehydration, loss of water, and it will make a bond like that. This is called a glycosidic linkage. Now, as I told, here you see that the carbon and oxygen, this bond and this bond, they are single bond. So, when we are thinking about the architectural hierarchies, what I mean by that, from where there are monomers, one unit, they connect to form polymers. Now, they would go on the higher level of structure. But if they are connected by single bonds, in between any conformation, remember I told about three secrets of covalent bonds, which are chirality, configuration and conformation. So, here they would adopt any conformation because free rotation is possible. That is the trick here, free rotation. And that is why the prediction would be very difficult. Polysaccharides are long chain of monosaccharide linked by glycosidic bonds. Different polysaccharides are connect through different type of glycosidic bonds. Let us take a look. So, this is, if you see, alpha 1 4 glycosidic bond, this is present in amylose, and you see that when it is alpha 1 to 4, this is forming straight chain. Now, when you look at another glycosidic bond, this is in cellulose and this is beta 1 4 glycosidic bond. So, as I told, once you see 1 to 4, they would be forming straight chain, be it a alpha bond or be it a beta glycosidic linkage. Now, you see the branching where you see the presence of both alpha 1 4 and alpha 1 6 glycosidic bond. This is present in a polysaccharide which is called as amylopectin. So, you see when you have 1 to 4 you get straight chain, when you get 1 to 6 you get branch. Number of such permutation and combinations are possible. This, along with the presence of a single bond in the connectivity, makes it nearly impossible to study carbohydrate. Forget about structure, even when you have to read the sequences for each and every bond, you have to use separate enzymes. Also, when they are complicated, for a same bond to get access, you need other enzyme. And this is very interesting because if you look at modern science is very much relying on utilizing what we call waste source. It is called waste to wealth. A lot of biochemical engineers, enzyme engineers, polymer specialists, they are involved in utilizing what we used to say waste before. Now they are trying to valorize them. And one of the classic example is lignin. If you get any waste, as an example, you could take any waste from the field where the crops are coming. Okay? So, let us say from the paddy field, from wheat, from corn, any waste you get a big amount of this as lignin. You will get cellulose, you will get hemicellulose and you get lignin. So, a lot of research is currently going on where people are trying to degrade lignin, because if you could degrade lignin, you could get a lot of value added product like coumarinic acid, synapenic acid and all. But because of the complex bonding they have, it required a lot of different enzyme, and what that is doing, it makes the involvement of multiple enzyme enhance the cost. When you are working with waste, you want the process to be cost effective, but it is not. Same problem actually happened in the field of carbohydrate everywhere. where we are not being able to get a sequence. So, the problem, I put lipid and fat here together because lipid and fat is not a polymer, but the problems now we are talking about is same. It become very expensive. More importantly, if you look at the polymers, the biological macromolecules, they have two very distinct difference. One, in one group there is DNA, RNA and protein. In other group there are carbohydrate, lipid, fat. What is the difference? If you see in the cell, there is chromosome. In the chromosome, there is genes which are made of DNA. The genes will tell you what would be the RNA or protein. So, what I mean, all the DNA, RNA and protein, they have a pre-made template, but in case of carbohydrate, lipid, fat, you do not have any template. Their synthesis is non-template based, and because of that, when you are investigating a particular cell, you have information about the chromosome, you know what type of DNA, what type of RNA, what type of protein you are going to have, but you are clueless about the carbohydrate and lipid. That is very critical. Non-standard, for DNA, you know that there are 4 nucleic acids where the differences happen. RNA is the same, for protein there are 20 amino acids, but carbohydrate, though we say that monosaccharide are forming polysaccharide, or in case of fat, glycerol and fatty acid, but there are number of fatty acids, number of monosaccharides which could be very different. There is no standard set for them. That is another problem, and they are very individualistic. They are very, or even inside a group of same organism, it depends on the food intake and many other things. So, what in summary I am trying to say, in case of carbohydrate, lipid or fat, first of all, even if you know about the organism, even if you know about the cell type, you still have no clue unless and until you do everything. So, there is no guessing. When there is no guessing, there is no prediction model. You know, a field could only be proceed further when you could develop a model for them. I know if this is coming, this would be this. Once biology gives you the opportunity of making rules, then only physics, chemistry and mathematics comes into and makes things develop in a proper way. Understanding goes into next level, which is a real problem in case of carbohydrate, lipid and fat. In case of protein sequencing, it is the practical process of determining amino acid sequence of all or part of the protein or peptide. So, remember we talked about this is the amino acid which is having NH3 and CO minus with a R here, R group on the top and hydrogen. So, if you see when one amino acid want to join with other amino acid, it would be I just make the charge taken out, so COH and NH2. So here again one water molecule, that is the fate of most of the polymerization where dehydration happen, and you get so this is the bond joint. Now apparently this is a, so first of all, what are the problems? Though this is the peptide bond, but these peptide bond behave differently, give different access. How the peptide bond could be? The first criteria is to break it, and the enzyme who are doing that is called protease. There are different type of protease, let us say serine protease, which works through the catalytic residue serine. There are different even serine protease, trypsin, chymotrypsin, elastase, a lot of them. Now, for all of these specified enzyme, they have different choices. So, again you have to use a combination, and when you have to make combination, this is costly. So, this may serve to identify the protein or characterize its post-translational modification. I did not talk about, but I will try to explain when I am going into the protein part. The protein, it is synthesized in the ribosome by covalently linking the amino acids, but after the process of synthesis, which is called translation, is performed, then there are enzymes in the cell, in the body, which could modify the protein. Like there are residues which are called having hydroxyl group, like serine, threonine, tyrosine. They could be phosphorylated. In that way, methylation, acetylation, there are post-translational modification. The two major direct method of protein sequencing are mass spectrometry. By the way, mass spectrometry is a field which have progressed a lot in recent time. So, I talked about the difficulty of carbohydrate and lipid, but using mass spectrometry and using high resolution NMR, it is possible to identify them individually. We will talk about. Mass spectrometry methods are now most widely used for protein sequencing and identification, but Edman degradation remains a valuable tool for characterizing a protein's N terminus. Sometimes, as I told, these are template based. So, you know a little sequence in the N terminus and you could get to find out the protein. That is done by Edman degradation. What is Edman degradation? Edman degradation is a method of sequencing amino acid in a peptide which was developed by famous scientist Pierre Edman. In this method, the amino terminal residue is labeled and clipped from the peptide without disrupting the peptide bonds between other amino acids. So, you could do that one by one. That is the beauty of the method. But it have a lot of problems. This process will not work if N terminus has been chemically modified. For example, acetylated or pyroglutamic acid is there, any modification. Sequencing will stop if a non alpha amino acid, non-essential amino acid is encountered like isoaspartic acid, since the favored 5-membered ring intermediate, which is essential for the process of this reaction to go on, is unable to be formed. Edman degradation is generally not useful to determine the position of disulfide bridges. Also, it requires high protein concentration. But beside the Edman degradation, even the normal sequencing using mass spectrometry is not very useful, because as I told, you have to digest, you need, the one problem is you need combination of enzymes. Second, individually the process is very expensive. So, this is not a process you could perform in a regular basis to the requirement of each and every protein you want to know. That is one of the biggest problem. So, we talked about carbohydrate, we talked about lipid, we talked about protein. The only polymers which are left are nucleic acids, DNA and RNA. We will come to that, but before that I would refresh your memory with the DNA research timeline, and I would include few new which would be extremely critical to proceed us in the right direction. So, first DNA research breakthrough was in 1865 with Gregor Johann Mendel. We know he is called as the father of genetics, and using the peas he did an extensive hybridization and developed basic laws of genetics. In 1869, Friedrich Misser, he come up with the nucleic acid which he called nuclein. In 1878, Albert Kossel from the same lab, Hopshire's laboratory, he come up with the name nucleic acid. He also identified that this molecule consisted of 4 bases and sugar molecules. In 1909, Russian-born American scientist Phobos Levine isolate ribonucleic acid and characterize the building blocks. In 1928, Fred Griffith carried out experiment on the pathogenicity of Streptococcus pneumonia using a virulent and an avirulent strain. His experiment would not be very conclusive, but that opened the field which was continued in 1944 by Oswald Avery, Colin MacLeod and MacLean McCarthy, who actually have taken the factor, add protease and DNase, and so that when there is DNase, the factor which is virulent factor is not effective, so it is DNA. Again in 1952, a more biochemistry oriented confirmation came from the famous Hershey-Chase experiment, where they use labeling of protein and DNA using sulfur and phosphorus and confirm that this is genetic material, this is DNA. Now coming to the new ones which I did not talk about. In 1950, Erwin Chargaff provide Chargaff's rule, which is in natural DNA the number of guanine unit equals the number of cytosine units and the number of adenine unit equals the number of thymine units. So, this is important definitely, but this was more critical I am coming to. In 1952, the real breakthrough come through the work of Rosalind Franklin. She studied the structure of DNA using X-ray diffraction. The first time people have used DNA to determine structure. She worked very hard to make a better and clearer pattern multiple time data collection, her discoveries indicated that DNA has a helical structure. This is Rosalind Franklin, and this is the famous pattern, and from this pattern they have even identified the distance between the two bases. In 1953, Watson and Crick, who were working, but they were trying to fit a model using Chargaff's ATGC rule, but they were unable. But by looking at Rosalind Franklin's work, it helped them to build a 3D model of DNA out of cardboard and wire. Their models were not correct or successful until they saw Franklin's X-ray patterns. Then they are able to build a correct model of DNA, which is double helix. So, this is Watson and Crick, and this is the famous DNA model they build up. When I am talking about DNA structure, I could not stop mentioning about my favorite scientist called Linus Pauling. Linus Pauling was a chemist, and that is definitely one thing I love about people like me who shifted from the field of chemistry to the field of biology. So, if you look at in 1931, Linus Pauling contributed to quantum chemistry with the work on the nature of the chemical bond, where he talked about the improvement of valence bond theory as well as the concept of resonance. If singly that would be contribution from one scientist, they would be happy enough. But Linus Pauling then, along with Robert Corey, discovered the protein structure using diffraction data. He was awarded with Nobel Prize for this work. He was not stopping even. The reason I want to talk about him here, we now know about the DNA structure. They are in A, B and Z form, and we know which form comes when and all these things. What is not mentioned regularly, the first model came from Pauling. This is the first model where he talked about the helical nature of DNA. So, the first breakthrough, the first concept was given by him. Only thing is, he was talking about triple helix, but it was proved that DNA is clearly a double helix, proving him wrong in this case. But today, if you look at, you will see that there are Hooksteen base pairs where there is evidence of 3 nucleoside forming interaction together and forming triple helix. So, it is not clear what he found that time. Might be he was unlucky to get not the regular DNA, but the Hooksteen pair. There might be many possibilities. Interestingly, this guy did not even stop there. He was also awarded Nobel Prize because of his contribution in peace. So, a rare scientist, a rare personality, but why I bring him here is because of the concept of helicity which came from him first time, which was critical to solve the structure of DNA. Now, we will talk about the basic properties of DNA. The base pair, which is a fundamental unit of double stranded nucleic acids consisting of 2 nucleobases bound to each other by hydrogen bonds. They form the building blocks of the DNA double helix and contribute to the folded structure of both DNA and RNA. Dictated by specific hydrogen bonding pattern, Watson-Crick base pair allow the DNA helix to maintain a regular helical structure that is subtly dependent on its nucleotide sequence. So, if you see there are 3 hydrogen bond between cytosine and guanine and 2 hydrogen bond between thymine and adenine. And here is the animation, you could see how they are forming. Coming to replication, DNA replication is the process by which DNA makes a copy of itself during cell division. The first step is unzipping. It is to unzip the double helix, because double helix is bound with non-covalent interactions and base stacking and hydrogen bond. So, unzip the double helix structure of the DNA molecule, and this is carried out by the enzyme called helicase, which breaks the hydrogen bond holding the complementary bases of DNA together. Now, when I am talking about this like incidents, one thing I am missing, and that is very vital in biology. Yes, helicase play the most important role to break it, but think about, you have a zipped molecule, you unzip it, there is another, like again a chance to re-zip. So, there are complex of protein specially called single strand binding protein, which binds to single strands after unzip, and in that way there are many protein comes and play their role to develop complexes which play very critical role, but most of the time we give the credit to the main one because it is so complex. Second is complementary base pairing. So, in complementary base pairing, the complementary nucleotides move into the position to bond with the complementary bases on the DNA chain. So, you see that this is the template strand and the bases are coming according to, if there is a it is T, if there is a G it is C, and vice versa. But if you look at a very interesting thing here, you see that the nucleotides are coming as triphosphate. So, they are coming as DNTPs and 2 phosphates are actually relieved. This is a very critical and interesting phenomena. If you think why they are doing that, take a look at the process of DNA synthesis. The nucleotides, many, so suppose there are 100 nucleotides, let us say, they are forming a polymer which is from 100 molecule to 1 molecule.